So what I want to do is I want to kind of give an overview of some of the work that we've been doing over the last several years related to this concept of give and take between host and pathogens and how it affects disease across numerous populations. So the real issue that comes up when you start thinking about this is what makes a pathogen pathogenic or not, because in fact, exposure to infectious agents is virtually ubiquitous, but infectious disease isn't. Most people who actually get exposed to a pathogen don't get sick, or often if they do, the episodes are mild, brief, and the exposure is effectively benign. So the question is, why is it that in a world where we're constantly battered by infectious agents, do we not always get sick? So I'm going to talk about two examples that we've been working on over the last several years. The first one is Helicobacter pylori, which is the causative agent for gastric cancer, much gastric disease, and duodenal ulcers. And the second example I'm going to talk about is Mycobacterium and the severity of tuberculosis. So to give a kind of a map from a paper we published last year, and this is for Mycobacterium, but it really occurs for most other things. You get an exposure to this agent, you get infected, maybe some people resist it, maybe some people get active disease very rapidly. But with the case of tuberculosis, most people stay in this stage their entire lives of latent infection with no active disease. So they carry the bacterium in their lungs. They don't transmit, at least we don't think so, and they don't get sick. So this is kind of a real norm, and I'll give you the numbers for Mycobacterium and tuberculosis because they're rather astounding how few people get sick relative to how many are in this latent infection stage. A similar scenario comes up with exposure to Helicobacter pylori and gastric cancer. So just to give you a kind of summary, Helicobacter pylori is the primary causative agent of gastric cancer. In 1900, it was the #1 cause globally of cancer death. It now currently accounts for about 10% of total cancer mortality worldwide. It's the third leading cause of cancer deaths globally. There are about 1 million new cases per year, and just over 750,000 deaths. Now, but if you can put this into perspective, more than 50% of the global population has an active H. pylori infection in their gut. There's significant genetic variation amongst populations, with the highest infection rate being in Africa. But the odd thing is, which was be a focus of this talk, is that infection rate does not necessarily correlate with disease prevalence. And that is, a question, why is a pathogen pathogenic? In fact, only about 1% of the infected people get gastric cancer, and about 10 to 20% develop ulcers. So to give you a little mycobacterial view, H. pylori is a curved, rod-shaped gram-negative bacterium with multiple flagella. If you look over here, you can see my cursor moving along the gut epithelium, and all these little black spots are in fact a colonized gut, and those are all the bacterium along the epithelial tissue of the gut. Now, it's kind of odd because in 1875, German scientists discovered that there was a spiral-shaped bacterium inhabiting the lining of the stomach. It couldn't be cultured. However, that became very rapidly forgotten. And for the next almost 100 years or so, people believed that the stomach was just too acidic to maintain a bacterial infection. Well, in 1979, Australian pathologist Robert Warren rediscovered H. pylori in the gut of ulcer patients. They published a series of papers, and they referred to the fact that there were these prior literature suggesting these bacterium existed. They did actually succeed in culturing it in 1982. It was odd because usually they were tossing their plates after 3 days. Marshall went away for a somewhat longer holiday, I believe it was Easter, and he came back 5 or 6 days later and there was growth on the plate. So they were slow growing. He also discovered at the same time that it causes ulcers by swallowing H. pylori himself. And he won the Nobel Prize, he and Barry Marshall and Robin Warren, for this discovery. So what happens is, if you look at a normal gut mucosa, which is odd because more than 50% of the people are infected. So by definition of this slide, more than 50% of the people don't have normal. So normal is probably an incorrect term, but you get H. pylori infection. There are a couple of genes in the Helicobacter, some of which CAG-A and VAC-A, that if you have specific variants, if you don't have the CAG-A and not all strains have that gene, you have non-atrophic gastritis. If you do have these, you can go down this cascade of gastritis, multifocal atrophic, and then down this cascade to cancer, or you can switch off to duodenal ulcer. And if you look at the histology slides, this is number 1 is the normal, and you can go out to the adenocarcinoma, which is 6. And these are scored by pathologists, and we use this information for our analysis. So basically, we are looking at patients who present with at least non-atrophic gastritis because they've actually been biopsied, and you don't biopsy healthy people for this. So the question is, what affects gastric disease risk or severity? And there are a large number of things. First of all, you have to be exposed to H. pylori. If you don't have exposed to H. pylori, you're not going to go down this pathway, or it's very unlikely. There's a host immune status. There's nutritional basis, if you're healthy or unhealthy in general. There's H. pylori genetics. I mentioned the CAG-A and the VAC-A genes affect disease. There's host genetics. But none of these factors that are written at the beginning really explain much of what we know about the disease. So we've asked the question, are there interactions between the host, not Mycobacterium, but H. pylori genetic interactions, and are these interacting in a way that are indicative of the two species evolving together to affect the disease presence and severity? Now, of course, I'll talk about the fact that we have to measure severity. So what is this concept of coevolution? And it's an old concept. It goes back in the evolutionary literature many decades. But it's really where two species reciprocally affect each other's evolution. They each increase their adaptation to the other species. And you can see this in spatial distributions and variation in spatial distributions. So one way of looking at this is there's a chronic pathogen, which is a prudent parasite, or it prudently exploits the host. There are many variables that affect the virulence, but one that we're going to talk about is this concept of vertical versus horizontal transmission. Vertical would be parent to offspring. In this case, it's mother to child historically for H. pylori, versus picking it up from the environment. Now, H. pylori, as I said, traditionally is a vertical, so that actually allows it to be very easily co-evolved because it's actually passed on from the mother to the child, much like mitochondrial DNA. But in certain environments where sanitation isn't particularly good, there is the potential for horizontal transfer. And there's a paper by Brights and Gagnon defining that coevolution between a host and a pathogen occurs when evolutionary changes in the pathogen then increase infectivity are counteracted by changes in the host. And this is really an evolutionary war going back and forth. So in our paper, we published about this whole concept of coevolution in about 7 or 8 years ago. The really is the point being that the effects of susceptibility and virulence are really contingent upon each other. So the host does something, the bacterium does it back, or the pathogen, and it just goes back and forth consistently. Similarly, virtually all of these kinds of coevolutionary interactions have very well-defined spatial variation. You don't evolve to a pathogen to which you're not exposed. So they have to be commensal, and they have to have adequate evolutionary time to adapt. We're seeing a situation where there is no coevolution now, right, with SARS-CoV-2. We're getting bombarded by a novel pathogen to which we have limited immunity. Now, if we were to look at this situation 1,000 or 5,000 years from now, it would probably look very different in terms of how the human is adapted to SARS-CoV-2. And this concept of coevolution is really difficult to prove because it involves an increased reproductive fitness in each species driven by the other species. And, this is done statistically by inference, as I'll show you. But it's really hard to demonstrate in real time. although it has been shown in some non-human species. So because H. pylori is vertically transmitted, so basically you're inheriting the strain that your mother had, who inherited the strain that her mother had, and so on, there should be this ability to kind of co-adapt to each other. And the other thing is that H. pylori is pretty well tolerated. About 75% of the people who are infected, so that's 30% of the world's population, have no noticeable symptoms, despite the fact that they have an ongoing infection. So H. pylori has coexisted with humans going back to the time when humans left Africa. And in fact, you can look at this map. The left-hand side is human migration out of Africa. The right-hand side is H. pylori migrations out of Africa. The one that we're going to talk about most today is actually the Middle Passage, which is predominantly due to the slave trade going back from the 1600s to the 1800s. And because the study population for this is in South America. So we've got this co-occurrence, so there's ample time to co-evolve. You've got this vertical transmission, oral to oral or to fecal, and you've got this very, very strong fidelity going. So why would we expect coevolution in general? Think about it this way. If the species co-evolve, both species survive. So if the pathogen kills all hosts, it no longer has anyone to infect. So therefore, it's an evolutionary dead end. So things that are really, really pathogenic will wipe populations out. Now, the other thing is that if the host eliminates all the pathogens, that's the end of the reproductive cycle of a pathogen. So there's this kind of arms race ongoing, which is really manifests itself as a coevolutionary adaptation. So, and the other thing is most infectious diseases really didn't hit humans till about 10,000 years ago with the changing climate and the development of agriculture, where population density went up, because you need a fairly high density to maintain an infectious pandemic. So here's the enigma. The prevalence of H. pylori infection in a population does not generally predict the incidence of serious clinical sequelae. It's a counterintuitive thing. You would think the more people that get infected, the more likely you are to see disease. And I said that the highest rates of infection are in sub-Saharan Africa. Well, that gives rise to what's referred to as the African enigma. And the same thing is true for certain Asian populations. So the frequency of infection and gastric cancer are not correlated. And to give you an example, this is from the same paper that I just cited before, Lunet and Barros. The populations, Bangladesh has over 90% infection rate, but only 1.3 gastric cancer incidents per 100,000. In Italy and Germany, it's less than half the infection rate, an order of magnitude more. And most of sub-Saharan Africa fits within this kind of general realm, being somewhere in the middle of 70 to 80 to 90%, with under 10 per 100,000. So there is this kind of disconnect, which is indicative of the fact that there's been this ability of the host to tolerate and the bug to tolerate the host. So these two enigmas are referred to as the African enigma and the Asian enigma. There's a third enigma, which actually is the one that I'm going to focus on for the remainder of this talk, where I'm talking about gastric cancer, and that's the Latin America high altitude enigma. It's been shown over and over again that populations that are at high altitude in South and Central America have a very high prevalence of gastric cancer, despite the fact that they have very similar infection rates to the lower altitude populations. And in the case of the study population, we're looking at the infection rates both in the coastal regions of Colombia and in the Andes are about 90%, but there's a 25-fold difference in gastric cancer incidence. So how do we study this? Well, we have to study case only. So because in fact, we don't know what an individual is exposed to determine whether or not they get disease. So with case only, we know what the strain is, so we can look at it, and we can look at genome by genome interaction in those people who are biopsied. And we can use disease severity as an outcome. And in the case of this study, we're using that cascade on a scale of 2 to 6, or we don't have gastric cancer in this study, but 2 to 5, and ask, what is the severity of disease relative to this interaction? Now, H. pylori is an incredibly diverse bacterium. About 6% of the genes are not shared between strains, so genes are just missing. It's highly recombinogenic when in fact multiple strains are exposed to each other, which gives the opportunity to break up and disrupt co-evolved complexes. So to give you an idea, this is ancestry of H. pylori based on sequencing of 7 housekeeping genes. Each vertical line is a single strain, and each color is a particular ancestry, and you can look at a given strain would have multiple mixed ancestries. For example, on the right over here, you've got Helicobacter pylori Africa 1, which is in the blue, is mostly from West and South Africa. You've got East Africa, where you've got this other strain, which is red. And then the European is predominantly dark green, and the Asian strains predominantly light green. But you can see that, in fact, if you take any strain out of the mix, any individual strain, and sequence it, you will say that it'll appear that it's not a solid single ancestry. So this allows us to say, well, how does the ancestry of the strain relate to disease? Okay, so this kind of enigma. And what people have studied, the genetics of the host, the genetics of the bug, as well as the environmental factors. And in fact, they all explain a little bit of the variation in disease prevalence and severity, but they don't explain very much. So the question is, neither the host nor the pathogen's genetic variation is very informative in our understanding of what the progression of the disease is like. So this concept of, because it's a recombinogenic bug, and in places like the New World, where there's been a huge influx in colonization of people who bring with them their own independent H. pylori that can then mix, which they have, you can disrupt these kinds of co-evolved complexes. And we can then use that information to test whether or not there's anything ongoing with respect to the story of potential coevolution and interactions. So in many parts of South and Central America, the incidence is much higher in the mountains, as I said. In our population, the H. pylori infection, our populations, the two we looked at, and I'll just show you where they are in a second, the infection rates are about 90%. But on the coast, the incidence is about 6 per 100,000. But in the mountains, with the same infection rate, it's about 150 per 100,000. So it's a 25-fold increase in gastric cancer incidence. So my colleagues who I've been collaborating with for a long time recruited 2 populations from 2 regions of Colombia. It was a natural experiment. They collected both blood from the host, and they did biopsies, and we cultured the H. pylori and then did the sequencing of the H. pylori housekeeping genes. We've now gone on to do whole genome sequencing and done a much more extensive both, but that data is not yet in. So the 2 regions we're looking at are in Colombia. It's this part of Colombia in the box here. Here's the Pacific Ocean. You can see that on the coastal region in Tumaco, these people, as I'll present the data, are a high percentage of African ancestry, whereas in the Andes and Tuqueras, the people are a high percentage of Amerindian ancestry. And these are the 2 populations that have this 25-fold difference in gastric cancer rate. So what we did is we used chip data for humans. We used, at this point, the immunochip was about 200,000 SNPs. We also did a GWAS, and we presented that separately. But in fact, we isolated paired samples from 233 people. We inferred ancestry from using the immunochip and structure, and then we used the sequencing that I showed earlier, the MLST, looking at the ancestry of the H. pylori. And if you look at the human ancestry on the coast, about 60% of the genome on average on the coast is of African ancestry, and in the mountains about 60% is Amerindian ancestry with European and mixed in. Very low percentage of European in both the coast and a moderate percentage in the mountains. The H. pylori ancestry, we see about 50% is of African 1, so the West and South African strain, whereas in the mountains, there's about 60%, 50 to 60% European. There is or was an Amerindian H. pylori. It has been pretty much supplanted by the European. And this is kind of a similar view to what I showed before, but on the left-hand side of it is the coast, on the right-hand side is the mountains, and each vertical line going down is the human ancestry, and for the same individual going down, it's the H. pylori ancestry. And you can look at this kind of cluster plot. Basically, the top half is the African 1 ancestry of the bug, the bottom half is the European, and along the x-axis is the proportion of Amerindian ancestry. So you can see as Amerindian ancestry increases for the humans, so does the European ancestry. And as Amerindian ancestry increases in the humans, the African ancestry decreases. And that is where we kind of got the idea to do this when we saw this plot. So we had biopsies on everybody who was over 40, using this Paleo-Carreo cascade of a scale of 1 to 6, and we simply did regression analyses. We also did ordinal regression, but I'll present the linear regression results because the results are virtually identical. So we used the histopathology score, basically that 1 to 6 score, as our dependent variable, as a measure of disease severity. And we looked at ancestry proportions, location, this kind of CAGA, this gene, which has been consistently shown to associate with risk of cancer. So again, here's this. So when we do that, the location, just testing for the location is highly significant, which is not surprising because there's a 25-fold difference. People who had a CAGA positive strain had worse disease, and all 3 human ancestries associated with histopathology. In the case of Amerindian ancestry, it had a positive beta, which meant the disease was worse, and African ancestry was less severe. And if you look just at the coast, the proportion of Amerindian ancestry was associated with more severe disease, slightly more than overall in the whole population. And remember that the bugs on the coast were mostly African ancestry. So when we look at it, we found that the H. pylori ancestry in and of itself did not associate with histopathology. And this is not unexpected if there's a kind of a coevolution. But when we looked only at the Amerindian population, we saw that the African H. pylori associated with really a fairly substantial decrease or increase in severity, decrease in mildness of disease, whereas the European ancestry H. pylori had a milder disease. So this was based on univariate analysis. We then actually did multivariate models to see whether or not these adjusting for all these other things, including an interaction term, impacted our model. So we looked at CAGA status, location, human Amerindian ancestry, African H. pylori ancestry, and the interaction between human and H. pylori ancestry. And we did that. We found that, in fact, that the interaction term was significant with a substantial beta. CAGA was unaffected, but the size of the beta was much greater for the interaction term. But what became really obvious and really interesting is that human Amerindian ancestry, which associated in a univariate model with a p-value of 0.002, lost all statistical significance. And location, which was highly significant, lost all statistical significance. So basically, when you adjust for this interaction, you lose the effect of the single variables. What does that mean? That means that the effect of the H. pylori ancestry in the univariate model was an artifact of its opposite effect in people who have African ancestry versus people of predominantly Amerindian ancestry. So what happens is the effect's not linear, and you see that African ancestry people in the mountains, did much worse. European did better. So what it appears is that people who are of Amerindian ancestry who are infected with the African strain do particularly bad because those are a strain to which they were not historically exposed, whereas people on the coast who were of African ancestry and predominantly had African H. pylori had a relatively benign presentation of disease. And you can see this kind of on our heat map with the exception of this little blip on the bottom left. This is the proportion of Amerindian ancestry for the humans, and the proportion of H. pylori. As you go up the diagonal, you get a worse histopathology score. So this is the significant interaction. This is a representation of the significant interaction term. So what we concluded in that paper was that host and pathogenic variation are more predictive when analyzed together than either one is alone. In fact, we really capture a much better picture of what the disease severity is being driven by. It's being driven by being infected with the wrong strain, or being infected with a strain to which your genome is adapted. Now, does this have clinical relevance? Now, remember, more than 50% of the people in the world are infected, so why don't we just go and treat everybody? Well, but the point is, can we figure out who's most likely to be treated? So we asked the question, the people who have the highest level of Amerindian ancestry at the 95th percentile, if they had a high level of African ancestry, H. pylori, they had a worse disease. Whereas if they had a low level, they had a more benign disease. Remember, higher number is more severe. And if you take the other end as people with a low percentage of Amerindian ancestry with a high percentage of African ancestry, H. pylori, they're doing okay. So there's the severity of disease is being driven by a discordance between the ancestry of the host and the bug. And now, so the question is, treat the people who have ancestries that are discordant. But it's not even that simple, because in fact, why do you not want to eradicate from everybody? And the answer is because H. pylori may be doing some things that are very good. First of all, if you eradicate, you increase the cases of gastroesophageal reflux disease and other symptoms, and you increase the risk of esophageal cancer. And we actually did a fairly intensive literature review this year, and we were able to show that although there are a whole bunch of things that H. pylori associates with that are deleterious, including gastric cancer and ulcers, they also protect from esophageal disease, asthma in children, inflammatory bowel disease, diarrheal disease. There's some evidence that it protects infection with H. pylori, protects from tuberculosis, food allergies, and celiac and Graves' disease. So we have a system where we can start assessing who do we want to actively eradicate H. pylori in relative to what are the downside of doing so, and relative to the upside if they're going to have a very severe disease and have a high probability of developing gastric cancer. So in conclusion to this part of the study, interactions between human and H. pylori genetic variation alter the effects of each other on disease severity, as measured by histopathology scores. There's evidence of a functional relationship between the two genomes. There's evidence for coevolution, probably due to this long-term coexistence with vertical transmission, and we think that this may aid in treatment decisions. So to go back to the African enigma, if you're infected, if you live in sub-Saharan Africa, you're most likely to be infected with an African H. pylori, which means you've co-evolved and you have benign or no disease, and there's the African enigma. Let me kind of transition now to the second part of my talk, which is the other, and these are not the only two species with which we've co-evolved, but these are the ones I've been studying most intensively. So Mycobacterium tuberculosis severity and coevolution. So Mycobacterium tuberculosis is the causative agent of most cases of tuberculosis. It's an aerobic acid-fast bacterium. It divides relatively slowly. Most lethal infections, it's the most lethal infectious disease globally. It kills more people than any other infectious disease until COVID-19. More than HIV, more than malaria. And it is predominantly in the developing world. But the funny thing is, despite how lethal it is, it's infecting a very large percentage of the people. 1 in 3 to 1 in 4 people globally are infected with Mycobacterium, but there are only 10 million cases a year. So why do most people who get infected with Mycobacterium not develop any disease? There are about 10 million cases, about 1.6 million deaths per year. It is the #1 killer of HIV positive people. So of the 1.4 in 2011, it's a little bit higher now, million TB deaths. About over 400,000 of them were also HIV positive. In sub-Saharan Africa, 75% of the people are co-infected people live. In Africa, greater than 40% of the TB patients are HIV positive. And in fact, one of the greatest susceptibility factors for developing tuberculosis on exposure to mycobacterium is being immunocompromised, i.e. having AIDS and being HIV positive. So if you're infected and you're HIV negative, there's about a 10% risk of the course of your entire lifetime of developing active tuberculosis, whereas if you're HIV positive, it's more like 10% per year. So there's a huge disparity in people who are immunocompromised. So it's extremely relevant in terms of what's ongoing in sub-Saharan Africa, where there's this dual epidemic. Now, but that said, most people who are not immunocompromised are going to be protected. There is evidence to suggest that there is some effect of the mycobacterium genome. There's some effect of the human genome based on both candidate gene studies and GWAS studies that have been going on for about 10 or 20 years. The candidate gene studies a little bit longer. But again, if you look at the host genome and you look at the pathogen genome, neither one does a very good job of explaining disease risk or severity. So what's going on? Well, clearly you have to have exposure. You have to have a healthy immune system in order to fight off and sequester the mycobacterium. There are genetic elements that have been identified, and there are clearly bacterial genetic elements, and the concept there are interactions. So in this study, what we're talking about is we're only looking at, again, with people with active disease. One of the reasons is because if you don't have active disease, you may have latent infection. In fact, there's a fairly high probability globally of being latently infected, but we don't know what the strain you have is, and we don't do those kinds of invasive studies, at least in general, to find out what people are infected with who don't present with disease. So again, it's a case-only study because that way we can determine what exactly is the strain you're infected with. And then we can ask, if you've got the disease, how severe is it in relationship to the genetics of the host and the genetics of the bug? So we assessed this kind of interaction the same way. Mycobacterium also left Africa with humans. They've coexisted for at least 20,000 years. There's some debate as to the exact timing of it. And the question is, if an interaction exists, does it affect the host and the mycobacterium specific effects? We saw that was the case with H. pylori. When you put the interaction term in, some of those other factors disappeared in terms of ancestry. Here you'll see that things appear. So can they modify to affect pathogenicity of mycobacterium? And again, we're looking at this concept of severity. This study was done, we again took paired samples and asked, is there evidence for mycobacterium genetics and severity? Is there evidence for human genetics on severity? Is there evidence for the interaction on severity? And how does that interaction affect the individual traits? So if there is an interaction, does it change the relationship between mycobacterium genetics and severity and or human genetics and severity? So this study was done in Kampala, Uganda, and part of the Coempi Community Health Study, household contacts. So everybody was either an index case or a household contact, therefore they had known exposure. We collected DNA from the hosts and mycobacterium DNA from the sputum of unrelated cases. We found that there were three lineages in our study. There's the L4 non-Uganda, which is there are 7 strains or 7 global umbrella strains of mycobacterium. L4 is the most ubiquitous globally. L3 is East Africa and Indian, South Asian, and L4.6 is a substrain of the L4, which is only found in Uganda and Rwanda, and maybe a few other surrounding countries. So it's been shown to be, of these 3 lineages, the most recently, i.e. the diverged lineage. So these are what we found in terms of the mycobacterium. How do we measure TB severity? Now, there are multiple ways of measuring TB severity, looking at x-rays and measuring the percentage of the lungs that are affected. We chose to do a method called the Bandem TB score, which was generated by a collaborator out of Guinea-Bissau. It's a measurement that is amenable to resource-limited environments. You're taking 11 variables of TB disease, and for the presence or absence of each of those, you get 1 point, so you can score 1 to 11. But we've also 2 of those, you can actually get extremes, and we gave extra points for those. And we've adapted that to our study in Uganda. So these are the variables: cough, hemoptysis, the chest pain. And then for BMI, if you have a BMI less than 18, you get 1 point. If you have a BMI of less than 16, you get 2 points. And mid-upper arm circumference of less than 220 millimeters, it's 1. If it's less than 200, it's 2 points. So you can vary from 1 to 13. The 2 cohorts we looked at were 2 cohorts sampled independently and genotyped independently out of Kampala. They totaled 234 people for this part of the study, 113 in cohort 1, 121 in cohort 2. They were similar with most regards, with the exception of cohort 2 had a lower HIV positivity rate, and so therefore we adjusted the model for HIV. In this first study, we examined the ancestry, which I said we found 3 of the mycobacterium, and we looked at 546 SNPs and we pruned them down to 395 SNPs and 29 genes in genes relevant to tuberculosis infection and response. The first thing over here is these are the 3 strains, and you can see that they're not different from each other, and the p-value is 0.68. So the lineage did not impact severity. Of the 29 genes that we looked at, only those in IL-12B replicated as marginal effects across the cohorts. There were SNPs in NOD1 and STAT1 that also associated in both cohorts. So we had associations in those genes in both cohorts, but there were different SNPs. IL-1B is one we've actually discovered with resistance to BB in an HIV-positive population, and it's been a well-studied, IL-12B rather, it's been a well-studied TB-related gene. So these are the SNPs in IL-12B that were replicated across the 2 cohorts. And here's our interaction model. So when we did the interaction model on all of our SNPs, and one of them came out as particularly interesting, this gene SLC11A1, which is formerly known as NRAMP1. It was the first gene discovered to associate with tuberculosis disease. In the model that included the interaction, the SNP associated significantly, the lineage associated significantly. Remember, in the last slide, I showed that there was no lineage association and the interaction was statistically significant. What I will say is that in the marginal univariate analysis, this SNP did not associate at all. So again, when you put the interaction term into the model, you effectively are stratifying by the different genotypes or lineages, and you're getting an effect. So the derived lineage, which is the L46 Uganda, associated with worse outcome, but only in the presence of the ancestral allele for SLC11A1. So basically, you have an ancestral allele that is pretty comfortable with the ancestral lineage to which it was exposed. You get a new strain, and all of a sudden, it doesn't do so well. So, but when we looked at this lineage, in the absence of lineage, the p-value for that SNP was 0.25, and here it's 3 times 10 to the minus 4. It's an enormous effect. And again, if you look at the literature looking at NRAMP1 or SLC11A1, as I said, it was the first gene discovered that associated with tuberculosis. But it's been really, really inconsistent in terms of the association. So I will leave the question with you, is that because the effect of that gene is MTB lineage dependent, and people did not look at the MTB lineage when they were doing these analyses. So in some studies it will come up as positive, in some studies it will come up not positively associating. So, but if you look at, I want to post, here's the issue. This independently was not associating, and you can see here, looking at the histogram, is that in the derived genotype, the Uganda lineage was better, but in the ancestral genotype, the Uganda lineage had a more severe disease. So you can see it's going down in this direction in this part of the slide and up in this. A classic example of an interaction. And if you look at it, derived versus ancestral for both the lineage of the mycobacterium and the alleles, it appears as if it's being driven by a coevolutionary phenomenon. So the mycobacterium lineage effects are host genotype dependent, and the host genotype effects are mycobacterium lineage dependent. Taken together, it really argues that coevolution is really what's affecting a predominant effect, has a predominant effect on the severity of tuberculosis. Okay, so we followed this up more recently with some recent data looking at a GWAS and looking at two cohorts, one of which is overlapping with the one I've just presented. We looked at one cohort, we did the Infinium Mega X, and the other we did the Illumina Omni 5M. We imputed to common SNPs of a few million. Again, the TB score differed between the two, as did the HIV positivity rate. Again, when we look at the same three lineages appear, when we look at them, we find that there's no effect of lineage in a univariate analysis whatsoever, which is not surprising. When we look at, this is a Manhattan plot, looking at all of the p-values for the interaction term using a 5 × 10⁻⁸ as a threshold, one SNP in one pseudogene, and I'll point out that it's a pseudogene, PPI-AP22, meets the bar of being genome-wide significant for the interaction term. So if we look at this, the SNP with the full model in the combined data is genome-wide, is very significant, 4 × 10⁻⁵. The lineage is significant when the interaction term is in the model. Remember, it was not at all significant in the model not adjusted for the interaction, and the interaction term is highly significant, indicating that we do have, in fact, evidence for a strong statistical interaction. We found 6 other SNPs that had a p-value of less than 10⁻⁶ and 91 to 10⁻⁵. And these are the other 6 SNPs that were significant at 10⁻⁵ or 10⁻⁶. So now, what I will say, it's a pseudogene, and here's the story going that, in fact, you've got the same going up here, going down here, exactly the same evidence for an interaction. This is a pseudogene, but this pseudogene is expressed in numerous tissues that are relevant to response to tuberculosis. And in fact, this pseudogene also associates with liver cancer. So it's a pseudogene, but it's not as if it's not doing anything, or at least it's not a linkage disequilibrium with something that's doing something. So in conclusion, infectious disease severity and perhaps presence is an outcome of both the host and pathogens genomes taken together. And we have to really, I stress the fact that this kind of coevolution is real, and it is probably much more pervasive. The third system in which it's been at least inferred and demonstrated to some degree is human papillomavirus. The disruption of this kind of coevolutionary relationship makes infectious disease worse. It probably drives susceptibility, but as I would point out, it's hard to know what an individual has been exposed to. And so what we argue is you have to add these interactions to models to reveal effects hidden by the marginal analyses. This was a lot of people working over many years, people at Vanderbilt. My graduate student, Nuri Kodeman, was the first author of the paper on the H. pylori. And this is the group that was working in Colombia. And then the work on Mycobacterium tuberculosis was being done with people at McCrary University, our host university, University of Pennsylvania, in Switzerland, and at my current institution, Case Western Reserve University. And I will stop there, and happy to take any questions.